Dr. Brent Coffey, director del Centro de Desarrollo de Aplicaciones Biomédicas, Laboratorio Nacional Frederick para la Investigación de Cáncer. Dentro de su labor, creó las reglas de algoritmos que se utilizan en el estudio del Instituto Nacional de Cáncer, conocidas como Matchbox, Sistema de Informática. Matchbox ha sido utilizada con éxito para evaluar las alteraciones moleculares de más de 6,000 participantes. Ahora sirve como el centro de sistema de informática, no solo en el NCI Match, sino también en el área pediátrica de Match y para próximas pruebas de medicina de precisión clínica financiadas por el Instituto Nacional de Cáncer. El Dr. Coffey fue reconocido por la revista Federal Computer Week por el éxito obtenido en su Matchbox como Rising Star. Ahora demos una calurosa bienvenida y un gran aplauso para el Dr. Brent Coffey.
Thank you so much for your kind invitation today. I've very much enjoyed seeing your beautiful country and your beautiful cities. My name is Brent Coffey, and I'm the Director of Biomedical Applications Development Center at the National Cancer Institute's Frederick National Laboratory for Cancer Research. My academic background is extremely varied. It's in business administration, and in biotechnology, and in bioinformatics, and in computer science. So today I hope that regardless of your scientific specialty, you will be able to take something of importance from my talk today. So like myself, at the Frederick National Laboratory, I'm in charge of a very large group and a very diverse organization. And I would like to ensure that I acknowledge them up front for their tremendous contributions to not my work, but to my entire group's work. So first of all, thank you to my CTRP and match engineers, and to my program managers. And thank you to my cloud and test engineers. And thank you to my security engineers, and to my bioinformatics and, next slide, bioinformatics and data scientists. And finally, I'd like to thank my sponsors at the National Cancer Institute for their continued support of my and my team's work. As an organization, my group's works on enabling clinical trials, oncology precision medicine clinical trials specifically. And besides bioinformatics projects dealing with oncology clinical trials, all of my organization's work have a few common informatics features. First, my organization specializes in developing enterprise-level systems. This means that our code is tested, is evaluated for security vulnerabilities, and is deployed in a professional manner. So while desktop scripts are often written in the field of sciences, and those are of great value, our software is a little bit different from those in that it's written to be supported geographically distributed environments by many people. It's meant to handle many error conditions and the rigors of a production environment. Second, my center develops cloud technologies, and we use leading edge technologies in this regard. And specifically, we use the AWS cloud. This allows us to have managed services and provides that AWS provides. And this allows us to concentrate on the solution instead of all maintaining hardware and software. Third, my center makes extensive use of a new technology, serverless computing. By this I mean that we use technologies such as lambdas that allow us to focus again on the solutions more than on resource and hardware allocations from operating systems. And fourth, we secure our data because we work with PHI and PIA data extensively. So we have data that is encrypted both at rest and in transit. But most importantly for my talk today, we work with artificial intelligence informatics in our efforts to cure cancer. So today I want to talk about one major project that we used artificial intelligence on and how this large project led us to create 2 demonstration experiments for our sponsors at the National Cancer Institute. Each of these experiments or demonstrations projects were based on the lessons learned in the previous. The first project I'd like to talk about is the NCI Match project. This trial is the fastest occurring oncology clinical trial in history and has been well publicized by many. But today I will talk about the AI component of this trial that evaluates the patients for treatments on this particular trial. After I discuss NCI Match, I will show how the lessons learned from NCI Match led us to receive funding for a demonstration project and experiment with scaling up NCI Match's AI components. And finally, I will show how that experiment or demonstration led us to this biomarker abstraction experiment. And from there, I will draw some conclusions about the future of AI in medicine. However, before I can begin, let me talk a little bit about what I mean when I am talking about artificial intelligence. In 1950, Alan Turing, who we know today as the father of computer science, proposed a concept that we also know today as the Turing test. Most basically, the Turing test is a simple way of explaining or defining artificial intelligence. And the Turing test postulates this. Blindfold a questionnaire. Have him or her ask a question to both a computer and to a human. If the blindfolded questionnaire cannot distinguish when they are getting answers from the computer or when they are getting answers from the human, then the computer can be said to be artificially intelligent. While the Turing test has its criticisms, the simplicity and the objectivity of the Turing test explanation and the fact that we still discuss it 70 years after shows some practical value, I think, to this simple definition. So the question for today is, as I move through my slides, the rhetorical question I would ask you to consider is, do the results that the computer give mirror the results that a subject matter expert may give? If so, perhaps we have a form of artificial intelligence. The other day, Dr. Feinbaum was on the National Institutes of Health campus. Dr. Feinbaum is considered the father of expert systems. And knowing I was giving this talk today, and I was already citing him here, I went to hear him speak. Interestingly, in his lecture, he not only cited Alan Turing, as I have just done, and frankly, as most do when they start a talk on AI, but he gave what I believe to be a clear illustration that described exactly what I will be explaining here shortly. So I present his graphic with a few minor modifications for the purposes of this talk and for this explanation. So while not the first to say it, he explained that artificial intelligence can be thought of as being composed of 2 parts, recognition and cognition. Recognition is the ability to identify information, and cognition is the ability to process information. With these concepts of recognition and cognition in mind, informatic systems can be seen as playing a role in artificial intelligence somewhere along the spectrum between recognition and cognition. Therefore, a good AI system is really a combination of informatic technologies, one part to provide the recognition capabilities, and the second part to provide the cognitive functions. Let us now see technologies that you may have heard of mapped along this continuum. On the far left-hand side, we see natural language processing, or NLP, which is really a very broad term, but here I specifically mean it in that it's primarily concerned with the recognition of words. If I see some characters, do I recognize those characters as something I am looking for? V600E, that's an amino acid change. Inhibitor, that's a particular drug function. Following, we see natural language understanding, which is often and really a subterm under the broad term of NLP, but which here is specifically in this context or is the understanding of a word or phrase. For example, if I were reading the eligibility criteria for a clinical trial, NLP may recognize V600E, but NLU would recognize if the molecular alteration in BRAF being written about is the eligibility criteria section of the protocol was inclusionary for a patient or exclusionary for a patient. So a complete NLP solution is one that has both recognizes terms and is able to understand the context of the terms in LU. Finally, under the cognitive side of the spectrum, on the far right-hand side, we see something called deep systems. Perhaps deep systems are what is most often discussed when someone is discussing AI in medicine. And deep systems are technologies that are like neural networks. And these systems are dependent upon results. Or in the clinical trial terms, these systems often need significant outcome data in order to be most effective, in order to be effective. However, also on the cognitive end of the spectrum is a deeply pragmatic technology, one that I believe is especially useful in precision medicine clinical trials that is not dependent upon outcome data. Expert systems. Expert systems, also referred to as rules engines, fall here on the spectrum. First, notice the graphic representation on this diagram as compared to that of deep systems. Expert systems are represented as small boxes as they are very good at evaluating specific domain criteria or decision trees in a manner that an expert could also evaluate, even if it took some effort by the expert. Let me explain a little bit more. Expert systems can be thought of as logic processing systems. Most basically, they take two inputs. First, expert systems take as input a set of rules. These rules are conditions of expert logic into the computer in readable ways, such as if-then or when-then constructs. These rules are developed by a computer scientist with a background in both computers and in the subject matter area in question. This person is sometimes referred to as a knowledge engineer. He or she develops these rules by interviewing subject matter experts or investigators in the case of clinical trials and or by reading the protocol documents. In this manner, logic of the system is built up, and essentially knowledge becomes represented in the computer. For example, a rule might be that a study participant must have an MLH1 amplification. And likewise, an ineligibility rule might be that study participants must not have adenocarcinoma of the colon. The second input to an expert system are facts. Fundamentally, facts are what is known and what the rules must evaluate. For example, a fact might be that a study participant has invasive breast carcinoma, and their tumor has a MLH1 amplification of 10. The facts are then merged with the rules to produce a result. And given the above hypothetical rules and given the above hypothetical facts, the study participant would be eligible for enrollment on this hypothetical clinical trial. Thus, we can see that in an ideal situation, the results are either true, the patient is eligible, or false, the potential study participant is not eligible. However, as we will see later, the outcome may also be indeterminate. And in trials, in many ways, this can produce the most interesting results. Expert systems were first developed in 1965 by Dr. Feinbaum, who I cited earlier, at Stanford University. His expert system was known as Dendril, and it was used to identify unknown organic molecules. To do this, it automated or codified the questions an organic chemist would ask when attempting to identify a molecule. In other words, it took expert logic and placed that logic into a computer. Expert systems then were famously used in 1972 in a project known as MySyn. MySyn was developed in order to provide consultative advice to physicians regarding the diagnosis and treatment of patients with bacterial infections. The reasoning behind MySyn was that many physicians were not experts at that time in infectious disease, and medical tests to identify the bacteria took several days. However, the physician needed to decide if the patient needed treatment then, and if so, what was the most appropriate treatment for that patient. MySyn was tested and shown to be effective. However, to the best of my knowledge, MySyn was never actually used with actual patients in a clinical trial. So coming back to the spectrum of AI informatics, here are the projects of Frederick National Laboratory for Cancer Research and where they fall along the AI spectrum. From this, we can see the various aspects of artificial intelligence are something that the Frederick National Lab and indeed the NCI is deeply invested in. As I've shown, expert systems are not a new concept. However, I want to talk today about how are we using this established technology, first used, as I pointed out, in 1972 in medicine, but using it today in production cloud systems with actual study participants entering precision medicine clinical trials. NCI match. In 2013, I was asked to work on a new precision medicine clinical trial known as NCI match. NCI match is a single finding trial conducted as a partnership between the National Cancer Institute and ECOG Akron. NCI match's goal, as Dr. Keith Flattery would write, is to find out if targeted therapies known to be effective in some tumor histologies would be effective in other tumor histologies for which the targeted therapy had not been evaluated. For example, if a drug had been developed to target MLH1 alterations in adenocarcinoma of the lung, will that same drug be effective in treating tumors from other histologies with MLH1 alterations? This trial would require enrolling patients to have a biopsy performed. The biopsy would then undergo molecular screening using Thermo Fisher's ion reporter, NGS, and Oncomine panel, as well as immunohistochemistry would also be performed. Once these laboratory assays were performed, data from these assays was generated in the form of binary alignment files, or BAM files, variant call files, VCF files, and then IHC's results would be sent to us as either positive, negative, or indeterminate. It is at this point that the assay results are uploaded to our system, which we call Matchbox. This is the point at which the NCI match trial began to use expert systems for the assignment of study participants to treatment arms. As I mentioned previously, an expert system is dependent on rules that are derived from experts, and Matchbox in this regard is no different. Therefore, Matchbox has a structured set of rules for each and every arm, as well as a common set of NCI match rules. So let us consider briefly the specific rules of Matchbox. Once the genomic assay data was uploaded to Matchbox, the first set of rules in place were to filter variant data. In this case, we run VCF filtering rules on a number of parameters. The rules we wrote included the following. Allele frequency is greater than 0. We checked to ensure the SNP was not intronic, and we checked also that it is not synonymous. If it's copy number our copy number variance that we're looking at, we ensure that the copy number is greater than or equal to 7. This has to do with our assays being calibrated for that particular level. And we also check to ensure that the variants were not known germline. There are a few more rules that are very specific surrounding read depth of particular variants, such as EGFR v3 as well. And these will be noted in upcoming publications. However, these I have shown give you a foundational understanding of how rules can be used to filter variant data. After we ensured that the variants were what we wanted to evaluate, Matchbox at this point would notify laboratory personnel that Matchbox has created something we called a variant report. This variant report was the result of the processing I just mentioned. At this point, Matchbox, running the rules, paused and waited for a bioinformatician to review the results and to sign off on those results. If the bioinformatician did not agree with the report in some way, they could reject the individual variants as being invalid, or they could request that variants be added back in to the report, or they could reject the report as a whole based upon quality scores. Of the over 6,000 variant reports created by Matchbox, 93% were able to be confirmed in this manner. But once the variant report was signed out, the rules would then pick up again and run the actual treatment assignment. Examples of rules included exact SNP and indel ID matches, protein change matches, copy number matches, fusion matches, assay matches that I've mentioned previously. And likewise, we would write exclusionary rules as well. For example, in fusions, if someone had an AGK to BRAF fusion, we might exclude them from an arm. But more interestingly, Matchbox could do something that we call non-hotspot rules. A non-hotspot match was something that was more complicated matching because it requires variant interpretation in some way. A simple example of this rule was if the variant report has a non-frame shift deletion in EGFR exon 19, then the patient is eligible. As one can see, this rule is not nearly as simplistic as a simple identifier match, but relies on metadata being accurately associated with the variant cause. Our expert system Matchbox is able to make such determinations by reading the annotation data in the VCF file. In addition to molecular alteration rules, there were rules written for each arm dealing with prior treatments of the patients. For example, patients who had previously taken nivolumab are not eligible for nivolumab arms. Or more complicated, patients who have taken an anti-PD-L1 are not eligible. Now, matching on drug names is straightforward, but I'll discuss the problem of matching on drug targets later. How does the computer know if a drug is anti-PD-L1? How does it know that? We will leave this question unanswered here in the NCI match, but we'll come back to addressing that with the following project. The results of running the rules for each arm then is that the patient is either eligible for no arms, for one arm, or for multiple arms. Yet our expert system must decide which is the single best arm for the patient. In the event a patient was eligible for multiple arms, our expert system would then run a set of rules we call tiebreakers. Examples of tiebreaker rules were developed as shown. First, we annotated all of our previous arm eligibility rules with something we called level of evidence, or LOE indicators. This was a simple numeric value from 1 to 3 that describes how likely it is that a patient with a given mutation was to respond to the prescribed treatment based on literature. More specifically, these were the levels. Level 1, gene variant credentialed for selection of an approved drug. Level 2, variant has eligibility criterion for an ongoing clinical trial for that drug. And level 3, loss of function, stop codon, models with variant response. respond, and without, do not respond. Thus, inclusionary eligibility criteria marked with 1 have precedence over inclusionary eligibility criteria with 2 or 3. However, this would not always break the tie between multiple arms, as they may have the same level of evidence. So the next tiebreaker was allele frequency. If we were comparing treatments between SNPs and or indels, molecular alterations with an allele frequency of delta greater than 15% were determined to be the better targets, and so they would be prioritized. After these and other miscellaneous tiebreakers were run, a tie might still exist. And in fact, it did exist in a rare occasion. And so a patient may be assigned to a lower crude arm, or if that still resulted in a tie, then the patient could be randomized between the arm. Assignment was always reviewed by an oncologist before being sent off to the patient. Before we opened Match, my experience in training and testing sessions we did with the experts was that early on there were many questions as to why an assignment was made by Matchbox. We would explain the computer's rationale for an assignment, as the assignment could become quite complicated with so many arms, and as you got into many, many details. I recall, though, that many long conversations with experts we spent significant time, and what we learned from this experiment, or from this experience alone, was that in fact, that we found that just building up the rules helped create a sense of understanding between all the physicians, which was also what they found when Mycin was created. Some experts have felt that encoding their knowledge into rules has helped them formalize their own view of the domain, leading to greater consistency of their decisions. In other words, the act of having to formally codify the rules and work with numerous arms and numerous PIs from many institutions using a rules engine led to great transparency. Matchbox has now been used in production to assign over 6,500 study participants to treatment arms with an accuracy of greater than 98%. So the rhetorical question I ask is, do Matchbox results match those that an expert would give? If Matchbox were to get an assignment incorrect, would it get the assignment incorrect any more often than an expert? We have to remember that the NCI Match trial was not designed to be an AI trial, and so it was not set up to answer these questions. It was a single finding trial. It was only once we started NCI Match and looking back on NCI Match that we began to think, we began to consider the questions that I present in these slides. So the creation of Matchbox forced us to ask questions about what is possible with expert systems. What would happen if we were to scale Matchbox up to work with any clinical trial or run by any organization? This would be useful for clinicians looking to find potential trials for patients. The ability to evaluate criteria different from current methodologies of finding clinical trials on websites such as cancer.gov or clinicaltrials.gov. Those systems work by searching for words or phrases. They don't actually evaluate the eligibility criteria. But if we could scale Matchbox, this would be a vast improvement over the current searching methodologies and greatly help save valuable clinician time. However, if we were to do this, we'd be faced with 3 challenges. First, recall that the drug exclusion, the anti-PD-L1 drugs, have various names and codes, just like diseases. So since Matchbox is being run as a centralized manner, Matchbox could have the input ontologies guaranteed. However, what if when we're trying to scale up an expert system to find trials run by different organizations where there was no central control or agreed upon data ontology? We would begin to solve this problem with multiple ontologies in this next study. Second, because NCI Match was a solitary trial, the criteria was standardized. While the totality of the criteria differed from arm to arm, as did the facts. Once the criteria was defined, it was consistent in subsequent arms with the same criteria. Now, this allowed us to more easily scale. Further, if a criteria was unclear, I could go talk to the PI directly. However, if we were to consider all open interventional trials, I could not as easily talk with the PIs, nor could I expect that the criteria of the trials would be standardized in any way. Thus, the manual creation of rules may be expensive, it may be extremely time-consuming, and it may prove to be inaccurate. And third, because NCI Match is a single trial with multiple arms, it could require that all data be entered for assignment. But what if I didn't have all the data for the study participants that I wanted to see? What if the doctor didn't want to enter all of the information? What if it wasn't available? Could they possibly be eligible for the clinical trial? Would an expert system be useful if something was unknown, so the answer to the if-then question was not true or false, but the indeterminate I referred to earlier? With those questions, we convinced our sponsors that Matchbox could be scaled up and led us to pros and receive funding for a demonstration or experiment project to prove that we could overcome those hurdles. We called this the Finding Cancer Clinical Trials Project. With the Finding Cancer Clinical Trials Research Project, I proposed a research project to scale up Matchbox to use an expert system in order to aid clinicians in finding cancer clinical trials most appropriate for their patient. This varied from our NCI Match expert system in that no longer were we using expert system to find an exact match for a patient, but we were using expert systems to narrow down the potential treatments for which a patient may be eligible by looking at the patient data that was available and the eligibility criteria that was programmed into the expert system. What you see on the slide, though, is my initial thoughts. This is a rough sketch that I wrote one day several years back on how we might scale up Matchbox. The goal was to greatly narrow down their search results, but to put the trial at the top with the best search results. In fact, the only thing that's important on the slide is on the far, which is your right-hand side, I believe, list of trials, as my inspiration came from BLAST, if anyone has used BLAST. It was simply to rank the trials in an ordered list and to score them appropriately. However, to define best, what does best mean? I divided the question into two parts. And we originally presented that there are two yet distinct, yet related algorithms. And each algorithm, should be done in distinct steps because they build on one another. First, which trial does my patient match? In other words, which trials does my patient meet the eligibility criteria for? The results of this algorithm would return all the trials for which a patient has eligibility, but sorted by a percent matching. Second, though, which trials are best for my patient? In other words, of all the trials my patient meets the eligibility criteria for, which are in the unique best interest of that patient? Perhaps phase of trial should be prioritized. Perhaps disease specificity should be prioritized. Perhaps geolocation should be prioritized. The results of this algorithm return the results of the first algorithm, but sorted in a manner most applicable to the patient's unique concerns. This algorithm would never return a trial that was not already returned in step one, but would narrow down the results of step one to a smaller subsection. However, we were funded to do only step one, yet step two's solution is readily apparent if step one is solved. As I mentioned briefly, the current methodologies of searching on cancer.gov or clinicaltrials.gov are simple keyword searches. In this current scenario, a clinician enters a keyword and has the website perform a match or partial match based upon the characters in a particular field in a database. For example, asking for all the breast cancer trials will result in the disease field in the database to pull back all the trials that have breast cancer coded in that field. This will result in a very large and unsorted set of trials that have breast cancer being retrieved. A clinician must now go through the eligibility criteria for each of these trials manually in order to find that one golden trial that's the right one for his or her patient. Some have proposed the way to solve this is by evaluating eligibility criteria strictly in a binary manner, true or false. Either a patient is eligible or a patient is not. However, consider a lab parameter, or consider an age, a 70-year-old patient, or other parameters that may change on a day-to-day basis. A strictly binary search may mean that the best trial for a patient may not even be returned. Our approach was to build an expert system to make a more intelligent search of cancer clinical trials and return a sorted list. Our goal was to remove clearly inappropriate trials and leave impossible trials, but most importantly, to score by likelihood of the study participants' eligibility. Since we had limited time and limited budget to build this demonstration or research project, we chose 207 trials from breast, lung, ovarian, prostate, and pancreatic cancer to test with. These 207 trials led us to create over 900 unique rules, but it only took us 6 weeks and 2 knowledge engineers to do this. Continuing, let's consider some sample criteria. Perhaps eligibility criteria might simply say no history of HIV, hemoglobin greater than or equal to 8, no chemotherapy in the past 3 weeks. By carefully considering the criteria, like the criteria I just showed, we realized that the mathematics for each criteria actually needed to be very different. Some criteria were indeed hard, true, or false. For example, history of HIV. Either a study participant has it or not. However, most criteria was actually a range, and that being a little below a cutoff perhaps shouldn't always rule out a study participant from being considered for that trial. For example, a lab score or a study participant's age may change in the very near future, and so perhaps ruling them out completely would not be the optimal approach of exposing the clinical trial as most appropriate for that patient. Other criteria more clearly only have short-term effects on a patient, such as chemotherapy in the past 3 weeks. And so ruling out a patient based upon that criteria alone does not seem optimal. Still, other criteria and other requirements were medical procedures to be performed. Yet some medical procedures are more invasive than others, and a study participant may be less likely to participate. The more invasive the procedure that a procedure is, the less likely a patient might be to want to actually do that, and vice versa if it's simpler. Finally, other items listed in criteria were really just notes and clarifications. To clarify the math or the range, let us look at this graph on this slide. Binary, as we see, is a hard true or false, which we see on the left-hand side. Whereas a range is a sliding adjustable scale that can either have a very steep drop-off, as this example shows, or could be more gradual. Our application allowed for this to be adjusted on a parameter by parameter basis if someone, a clinician, so chose. However, using the mathematical categories and working with our experts and our knowledge engineers, we came up with a default weighting criteria. And by default, all binary criteria had to be met, and we gave range 90% of the weight, and short term 10% by default. However, all of our defaults are completely customizable by users of our application. In fact, we expect and we wanted clinicians to adjust the weights for their study participants' unique needs and adjust them as they saw fit. But finally, we had to deal with missing criteria. We realized that criteria could be missing for one or two reasons. Either data was unknown, or perhaps the criteria did not have a rule created for it. However, with this application, we weren't trying to assign a study participant to an exact particular trial, but to score trials, we realized that missing criteria should lower a score, but it shouldn't rule out a patient as being ineligible. And finally, as I just mentioned, eligibility criteria may not have a rule written for it. Well, there's a reason for that. This was because it became quite clear that abstracting all criteria from a trial had a diminishing return, and we had to weigh efficiency and performance against perfect accuracy. Since in the case of finding cancer clinical trials, we weren't trying to assign a trial, but find a list of potential trials, perfect accuracy was unnecessary, as the clinicians would not want to enter all information on study participants, and some criteria was so specific that it would rule out only a minuscule number of participants anyways, so it simply wasn't worth it to outweigh that over performance. As expected, as we moved through the process of creating the rules for 207 trials, in our test set, we found that the criteria was repetitive, and that it was only really the facts, the specific value of the criteria that was changed. Therefore, by the time we got to trial 200, we found that we weren't writing rules any longer, that we were simply changing the order of the rules and changing the facts. Here's a screenshot of our completed demonstration project. As you can see, the expert system was able to use our algorithm to weigh the criteria based upon the input and bring back a sorted list of scored trials. Further, clicking on the trial shows clinicians the reason for the score as it lists the eligibility criteria and indicates clearly what matched, what did not match, what data is missing, and using the range algorithm that was close to matching but did not. Recall also from NCI Match that I mentioned anti-PD-L1, that drug challenge for us. Our Finding Cancer Clinical Trials application demonstrated that an expert system could be connected to ontology services in real time to evaluate rules with different naming conventions. Here on the right, we see that the eligibility criteria stated simply no steroids within the last 7 days. And on the left, we see that the patient information was dexamethasone as having been taken in the last 7 days. So the expert system understood that dexamethasone is a steroid. So when we consider the challenges of NCI Match that NCI Match presented, we see that having study participants' data come all using all the same standards isn't completely necessary, as long as the participant data is using some knowledge, some known dictionary, as the expert systems can be linked to ontology services, but only a single rule needs to be written. Next, we see that standardizing protocol eligibility criteria is a nice to have in NCI Match, and it made things simple and easier. However, even without the uniformity of expression, we were able to quickly build up a library of rules that could be reused over and over again, even though the trial's eligibility criteria was written by different people and different organizations. And finally, and perhaps the most interestingly, we see that modern expert systems can handle missing data. We see that we can use expert systems to in creative ways to use unknown data values and still get interesting, useful results. So the rhetorical question I ask again is, does the expert system in finding cancer clinical trials match those that an expert would give? If the finding cancer clinical trial expert system were to get order of trials incorrect, would it get it any more incorrect than an expert? Well, we believe that our little short experiment shows that expert systems could be of significant value for finding cancer clinical trials, even with missing data, even with missing eligibility criteria, and even with different ontologies. This final piece of the puzzle, though, brings us to our last experiment. I refer back to my original sketch of what finding cancer clinical trials might look like if we had used NLP and NLU for the creation of rules. Given budget and time constraints, we were not originally allowed to test out the NLP and NLU parts. However, later, abstracting biomarkers from clinical trials was becoming more and more of a concern for new trials that we were getting ready to do. So we were asked to follow up with our project and demonstrate NLP and NLU in understanding biomarker eligibility criteria specifically in precision medicine clinical trials. So finally, let us consider the demonstration of biomarker abstraction and protocol eligibility. We pick up where we left off and circle back to NCI match. And we look at the protocol documents and some other internal documents to see if we could use existing NLP packages to correctly recognize and understand biomarkers in the context of match. Listed here are some of the tools we experimented with. While I will not show all the results, I will show the best two. After scanning 157 documents into our NLP pipeline between Seth and TMVAR2, we consistently got a 90% success at recognizing variants in our protocol documents before training of any of the packages. After training of the packages, we were able to get over 97% success at recognizing variants. However, recognizing variants is only half of the issue. We still have to understand the context of the variant. Is it inclusionary? Is it exclusionary? Or is it something else completely? Recall, recognition is only half the issue. We must determine if the computer understands the context in LU. Two example statements from the NCI match protocol documents are shown here on this slide. Notice the statement in blue is an inclusionary statement. I also notice it refers to Appendix 4 for a specific mutation that defines the specific molecular alterations of inclusion. The second is a statement of exclusion, but is not specific on what is meant by activating KRAS. This lack of specificity leads to some difficulty in the computer understanding the exclusionary intent. However, even with the difficulties I noted, when we took into account correctly contextualizing the variants, we see that we received 86% accuracy with reading the corpus of 157 match protocol documents. So the rhetorical question I ask is, the use of AI systems results match those that an expert would give or might give? The bioabstraction NLP expert system was a short and not a fully fledged project by any stretch of the imagination, but it does help us understand the state of artificial intelligence today. When would it be useful? Where does it lack? And how its shortcomings may be ameliorated. Our initial testing of NLP and LU to pull out clinical trial biomarker eligibility criteria was promising. While we cannot say that it will be 100%, we do believe that this initial sketch that is on the screen, which shows NLP being used with a person reviewing the criteria and the biomarker, is a viable plan. A plan to assist AI with a human should reduce the amount of work required to be done manually, but by having the person involved, the accuracy of the data abstraction can likely be increased. This observation leads me to my conclusion about the near term and the future of AI in medicine. So, from the NCI Match project and the 2 demonstration projects, I believe we can draw these conclusions. First, our experience with the expert systems has shown that they are mature and viable means of using artificial intelligence today in a production environment. Further, our experts have shown that expert systems and the use of AI technologies, when used together like NLP and NLU, could provide even greater value to cancer research community in the future. And in fact, the National Cancer Institute largely agrees and has funded the Frederick National Lab to continue using expert systems in increasingly advanced ways with the introduction of 3 new upcoming precision medicine cancer clinical trials: AML match, immunomatch, and combo match. However, when it comes to the near future of AI, it is my personal prediction that the best use of AI and informatics is an important and indeed critical aid or assistant to clinicians that is capable of bringing the greatest value. So just as in Greek mythology, the centaur or man-horse combination made for great stories, something we call centaur systems or AI-assisted systems or clinical recommendation engines, I do believe will continue to grow in usage and popularity for the near future. This will greatly benefit the more advanced usages of AI in the future. Thank you.